Now we will study components of a cell. Cell wall. Cell wall is present only in plant cells. Cell wall is made with the help of carbohydrates like cellulose and pectin. It gives protection and support to the cell by not allowing excess of water into the cell. Plasma membrane. Plasma membrane is present in plant cell as well as animal cells. Plasma membrane is a thin, fragile and elastic covering of a cell. It does not allow all substances to enter inside the cell. It is a selectively permeable membrane. As a result of it allows oxygen, water molecules and salt into the cell and exits carbon dioxide from the cell. It is made with the help of two layers of phospholipids and protein molecules are embedded in it. Embedded means deeply fixed inside the phospholipid layers. Cytoplasm is the fluid matter which is present in between nucleus and plasma membrane or cell membrane of a cell. Inside the cytoplasm, cell organelles are present. Cytoplasm is having the capacity to move from one place to another place, and it is a sticky substance. Other than cell organelles, a fluid matter is present inside the cytoplasm that is known as cytosol. Cytosol. Thus, each and every cell organelles has their own lipoprotein covering or membrane. Other than nucleus and chloroplast. Nucleus and chloroplast. Other organelles can be seen with the help of electron microscope only. Inside the cell, number of organelles are present. For seeing nucleus as well as chloroplast, electron microscope is not necessary. At the same time, remaining organelles can be seen only with the help of electron microscope. Cytosol stores different nutrients like amino acid, glucose, vitamins, etc. in it. Nucleus is the most important organelle of a cell. It consists of a double-walled membrane. Inside the nucleus, there is nucleolus and chromatin fibers. At the time of cell division, chromatin fibers condenses to form chromosomes. Each segment, or we can say that each functional segment of a chromosome is known as gene. Nucleus controls all metabolic activities of a cell and it helps in cell division. It transmits all hereditary characters from one generation to other. That means from parent to offsprings. Endoplasmic reticulum is like a network, or we can say network like structure. It has small, or it is made with the help of small tubes as well as flat sheets. In these tubes as well as sheets filled with fluids. It has connection from nucleus as well as plasma membrane. Inside its connection is with the nucleus, and outside it is having the connection with the plasma membrane. It conducts nutrients from one part of the cell to the other part of the cell. There are two types of endoplasmic reticulum. They are rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum has ribosome grains in its outer surface. Smooth endoplasmic reticulum does not have grains of ribosomes. They appear different. That means rough endoplasmic reticulum and smooth endoplasmic reticulum looks different, even though both of them are the parts of the single endoplasmic reticulum. Golgi complex. Golgi complex consists of flat sacs. Each sac is parallel to each other. The protein which is coming from endoplasmic reticulum enters in the vesicles, and vesicles moves through the cytoplasm and reaches to the Golgi complex. Now each sac of the Golgi complex is known as cisternae. Cisternae. Its structure almost like that will be there. Each one is parallel to each other. Even they are connected to each other. This phase, one phase is known as formation phase. The other end or other surface, that is known as maturation phase. The vesicles collect protein from the endoplasmic reticulum and reaches to the formation phase of the cisternae. It reaches to the formation phase of the cisternae. Then, as and when vesicles reaches here, these proteinaceous substances enter into the cisternae. Inside the cisternae, there are enzymes. These vesicles bring proteins. As and when proteins enter inside the Golgi complex, some chemical reaction will take place inside the Golgi complex. Then those material will reaches to the maturation phase. Here again vesicles will come and collect the materials which are forming, or as a result of chemical reaction which product is forming, that product will collect by the vesicles at the maturation phase and they do the distribution of the nutrients in each and every part of the cell. So here we can say that at the formation phase, proteins will enter inside the Golgi complex. Inside the Golgi complex there will be enzymes as well as proteins. Some chemical modification or chemical reaction will take place. As a result of it, their new product will form. That product will take by vesicles and they distribute those nutrients to each and every part of the cell. So we can say that Golgi complex has packing as well as distribution work. That means it takes, so we can say that packing work. Afterwards, once it is chemically modified, then it passes to the vesicles which are coming near to the maturation phase. Afterwards, we can say that these nutrients distribute each and every part of the cell. Golgi complex does different functions in the cell. Golgi complex is the secretory organ in the cell. It modifies, sorts and packs materials synthesized in the cell. It dispatches or distributes those synthesized materials to the plasma membrane, lysosomes, etc. Golgi complex produces vacuoles and secretory vesicles. Golgi complex helps in the formation of cell wall, plasma membrane and lysosomes. Lysosomes. Lysosomes are simple single membrane bounded sacs. Due to various metabolic activities, organic waste is generated inside the cell. These organic waste digest by lysosomes. Lysosomes performs various functions in the cell. They have big role in the immune system. They destroy viruses and bacteria which attack the cells. They are known as demolition squads because they destroy worn out cellular organelles and organic debris. They are also known as suicide bags. When old and damaged cells will be there, lysosomes burst open and enzymes digest their own cells. During starvation, enzymes which are present in the lysosomes digest the proteins and fats present in the cells. We can see mitochondria only with the help of an electron microscope. Mitochondria contains a double-walled structure. This is known as outer membrane. Inside this outer membrane, you can find many folded inner membrane. This is known as inner membrane. Inner membrane has many such folds are present. Each fold is known as cristae. Inside this membrane, a proteinaceous gel-like matrix is present. So inside this inner membrane, a proteinaceous gel-like substance is present, that is known as matrix. Now, inside this matrix contain ribosomes, phosphate granules, and DNA. With the help of ribosomes, phosphate granules and DNA, protein is produced inside the mitochondria. The enzymes which are present inside the mitochondria can oxidize carbohydrates and fats. As a result of it, energy is produced inside the mitochondria. The energy produced inside the mitochondria is stored in the form of ATP molecules. In the form of ATP molecules. In plant cells, mitochondria is very less, but at the same time, in case of animal cells, number of mitochondria is more. Mitochondria produces ATP molecules in the cell. ATP molecules are the energy rich compounds. With the help of ATP molecules, mitochondria produces carbohydrates, proteins, lipids, etc. in the cell. Vacuoles are the storage sacs for liquid and solid contents. They are bound by a single membrane. Vacuoles do not have a typical shape or size. Their structure changes as per the need of the cell. Vacuoles perform different functions in the cell. Vacuoles maintain osmotic pressure in the cell. They store metabolic byproducts and end products. In animal cells, they store waste products and food, while in amoeba, they store food before digestion. In plant cell, they are full of cell sap and provide rigidity to them. Turgidity means it will appear in the swollen condition. Cell will appear in the swollen condition, and rigidity means because of the help of vacuoles, cells will be appear more stronger. Chloroplasts present only in plant cells. They are having double-walled membrane. Even they are having oval-like shape. It has two walls. Inside the wall, stack of thylakoids are present. We can say that coins kept one above the other. Like that way structure is there. Such a way coins kept one above the other. Like that way structure is there. Then we can say that they are joined with each other. Like that way they are joined with each other. Now we can say that each one, whole thing we can say, all together we can say as granum. Single one we can call as thylakoid. Joint between each granum, this is known as lamella. The space between granum as well as, we can say that this inner membrane, we can say that is stroma. Now inside this stroma having ribosomes, DNA, enzymes, carbohydrates. So inside the stroma there will be ribosomes, DNA, enzymes and carbohydrates. These substances are necessary for photosynthesis. Main function of chloroplast is photosynthesis. Inside the chloroplast, a pigment is present. That pigment is known as chlorophyll. This chlorophyll helps to absorb solar energy and convert to chemical energy. Chemical energy has got one more name, that is food. The chemical energy itself is the food or the food material which is preparing by the plant cell. So inside the plant cell, number of organelles are there. We have studied in detail about each and every organelles. Chloroplast is the last organelle. It is made with the help of double-walled membrane. Inside this membrane or double-walled membrane, we can find granum, lamella as well as stroma. Here this stroma contains ribosomes, DNA, enzymes as well as carbohydrates. Inside this granum there is chlorophyll. With the help of chlorophyll, plant cells can absorb solar energy and convert that solar energy to chemical energy or food. Thank you.